So today I'm going to present virtual search of natural SARS-CoV-2 inhibitors among natural products with dual anti-inflammatory and antiviral activities. This work is just a perspective of drug discovery against COVID-19. As you can see, my name is Sandjo. I'm from Cameroon, and I did my PhD over there, and later on I finished my PhD in France. And what else I can say? As maybe some of you guys know, Cameroon is in central. It's a country located in Central Africa, a little bit above of the Ecuador. And so I was in Brazil in 2011 for a project, and then I visit Florianopolis and other cities, and later on I got an invitation as a visiting professor at WUFSC, and from there, from 2014, I decided to install myself in Brazil. So I was recruited here at WUFSC as a permanent professor. So let's continue. COVID-19 is a new disease caused by a virus called SARS-CoV-2, which put the entire planet in the pandemic. So the outbreak was announced in December 2019, which led to social isolation and distancing. And with the use of masks in the entire planet. So the evolution of the diseases was characterized by massive transmission and infection, emergency care unit overcrowded, and international lockdown. So you have a lot of healthcare system which was out of control because they received a large number of patients, and you also have a kind of prohibition to travel by airplane from a country to another. So because of this, there was a kind of evolution to respond, let's say, to fight these diseases. And this response was typically from the scientific communities. But the first idea, beside the prophylaxis, the first idea was drug repurposing. So this possibility did not work. It failed. And as you may know, there are many drugs which were trying to combat SARS-CoV-2, such as chloroquine, hydroxychloroquine, azithromycin, remdesivir, lovinavir, ritonavir. But none of these drugs was able to increase the survival of some patients. So after some meta-analysis, there were no change between the patient using this drug and the patient which are not using this drug. So this possibility failed. And the second one was let's say a race to develop vaccine, which I will say is quite successful because some countries such as the United States you have some states there which are not they are not using any more masks, and neither using social distancing and isolation. And you have a bunch of vaccines which were developed, such as CoronaVac, and the Pfizer vaccine, the AstraZeneca vaccine, the Moderna vaccine, and so on. Okay. But beside this, let's say, temporary solution, we can notice that some of the countries, let's say the tropical countries, which are not really rich countries, are still out of control. The social distancing is not working. The social isolation is not working. And you have the occurrence of SARS-CoV-2 variants, which high transmissibilities and virulence. And the consequence of all this, let's say, situation is the collapse of the healthcare system in various countries, the increase of the death rate, the economical and social impacts, and the emerging of eradicated diseases. As, for example, in Brazil, if I remember, you have there were many neglected diseases under control, such as tuberculosis and multi-drug resistance infection diseases. But now, with the social lockdown, there is no possibility to communicate between, let's say, the consortium responsible for this control with the population which are affected. You can see the case in Manaus, where people which are really far from the city, they have some diseases such as leishmaniasis, Chagas diseases, or malaria. And the control of all this, chikungunya, or dengue, the control of all these diseases is not possible anymore. And then you have many co-infection occurring, and this has a great consequence with the increase of the let's say victim. And based on all this observation, this is just a unique case of Brazil. But if you can think about India, you can think about some African countries. The situation is, let's say, nearly the same. Although you also have some political responsibility, but we will forget about it because it's not relevant. And then, based on this observation, there is a huge demand, not only for prophylactic solutions, but also for corrective solutions. I will not, let's say, I will not start the main work without raising the symptoms. Some of the some observation which were made was the symptoms goes from fever, cough, shortness of breath, and dyspnea to severe acute respiratory syndromes. And the symptom grade is different from individual to another. You have some infected individual which are asymptomatic, and other present severe pneumonia and SARS and organs failure, even death. And moreover, blood analysis of hospitalized COVID-19 patients revealed that there is a state of out of control level of numerous inflammatory mediators, which means in their blood there were many chemicals which are let's say a consequences which induce inflammation. And besides this, there was also observed a dysfunction of the immune response in patients with comorbidity or those facing severe symptoms and hyper-inflammation response. So, based on all these symptoms, we noticed that before before COVID-19, there was also a disease, I think 10 years ago or more than 10 years ago, which similar symptoms. It wasn't that, let's say, lethal like now, and it was only in Asia and in the Middle East. So our hypothesis, and this disease was caused by MERS and SARS-CoV, and then our hypothesis is what can we, let's say, what can we use from SARS-CoV, or what can we learn from SARS-CoV that are important to develop something to fight SARS-CoV-2. So the first thing is, a study show that a molecular biological studies show that SARS-CoV-2 and SARS-CoV has 96% of similarity on their cysteine protease. So the sequence was analyzed, and they found out that you have 96% of similarity. And about concerning papain-like protease, both viruses presented 86.5% of resemblances. So they are quite a good starting point from this study. And then morphologically, both viruses have a single-stranded RNA protected by an envelope formed by a membrane, which bears spike proteins. So this is a second similarity. Based on this similarity, we found that, okay, if we have some natural product which can inhibit SARS-CoV proteins, maybe, or based on the fact that both viruses have some similarity, are almost similar on two, let's say, two main proteins, maybe natural product which can inhibit this enzyme can be used as a starting point to fight SARS-CoV-2. So we found that numerous natural product was previously reported, identified from medicine plan and reported with these activities, and therefore SARS-CoV and SARS-CoV-2 are almost alike, and since they are similar, so inhibitor of SARS-CoV proteins are maybe suitable alternative to develop anti-SARS-CoV-2 agent. So why focusing on natural product? First, natural product have multi-functionalities. Second, they have structural diversity, which give them the potentiality of multi-target, or let's say they can target virus pocket in a protein, or they can also target different type of protein at the same moment. So this can be an advantageous to restore the immune response, to control the infection, and to control the hyperinflammation. So based on this, we did a literature search. The first thing was, we found in the literature natural product which were tested on SARS-CoV-2 proteins, and among them 79 plant metabolite was able to inhibit at least one protein of SARS-CoV, and they also have some anti-inflammatory properties. So 79 compound was able to show these two biological properties. And among them, among these 79, 14 was found to be inhibitors of this enzyme, and they were able to modulate the immune response. So as a result, I will show you some structure here that we found in the literature with those activities. The first one is amintoflavone, and I guess he's the most active one. No, it's the second active compound behind myricetin. So you have also apigenin, espiritin, camphorol, quercetin, and luteolin. But if you notice, below the name of each compound, I indicated the sources. All of them, as you can see, it's a good selection I did here. You also have some natural product. I couldn't put all the structure because it should have given a lot of page to be talking about. But I just selected the most interesting in the way that they are from edible plants. You can see that amintoflavone is from pistache, apigenin is from celery, epiritin is from citrus fruits, lemon, orange, and so on. Myricetin is from grape and blackberry. Camphorol is from apple, asparagus, broccoli, and onion. Quercetin to apple, grape, and onion. Luteolin, onion, broccoli, and oregon. So this is also another, let's say, another way to observe him, and this can raise, let's say, a very good question about what is the role of functional food with COVID-19. But we didn't go that side. It's just a question that I'm raising here. In the second plan, we checked the anti-inflammatory activity of all those molecules. You can see that they are quite interesting. Amintoflavone, for instance, can inhibit chemosine like N-oxide and cytosine like interleucin 1 beta and tumor necrosis factor alpha. And even though all these cytokines was found in different kind of cells, at least it's already a good starting point. They have their anti-inflammatory activity reported. But you will see at the right part of the slide that I highlighted two structures, namely epiritin and quercetin. Why? Because epiritin, for instance, show anti-inflammatory on post-myocardial infarction in mice. So, based on the literature, after administering epiritin on mice which suffer cardiovascular diseases, they were able to increase the survival of the animal. So this is a good, let's say, a suitable molecule to be used as a drug candidate. Quercetin also shows anti-inflammatory activity on lung cells. And after I will explain more why I chose those two, why I highlighted those two molecules. But what you can keep now is one, show anti-inflammatory activity on mice with post myocardial infarction, and the second one show anti-inflammatory activity on cytokines which were produced by lung cells. And the other class of compound which shows inhibition on SARS-CoV protein with anti-inflammatory activity was isoflavonoid, namely psoralidin, and you can see that it shows a quite good activity on papain-like protein, and it was able to reduce the level of inox, which were produced in RAW264 inflamed cells. And also it shows some inhibition effect on chemokine and cytokines in human normal lung cells. So that is why I highlighted this molecule, because once again his anti-inflammatory activity was against inflamed lung cells. You also have neo-bava isoflavones, which show, let's say moderated inhibition effect on papain-like protein with inhibition effect on chemokine and oxide production and cytokines such as interleukin 1 beta, interleukin 6, interleukin 12p14, and interleukin 12p17. You also have a granulated isoflavonoid with a moderated activity also, and dasein. Dasein is isoflavonoid found in I think it's soja, but his activity was quite weak, even though he shows anti-inflammatory activities. Another group of compounds which were explored was diterpenes, and namely cryptophencinone, inoculol, furogenol, abiotic acid, and dihydrotensionol. And as you can see, they show quite good and weak activity respectively against PL-Pro and 3CL-Pro. And the inoculol show activity only, it was tested, what we found is only on the 3-cysteine protease. And the furogenol was only also against 3-cysteine-like protease, as well as abiotic acid. But dihydrotensionone was evaluated against both, and it showed quite good activity against these two proteins. If you compare with compound 12, you will see that his activity is quite good against both proteins. We also find some diaryl ethanoids with good inhibition effect against 3CL-Pro and PL-Pro of SARS-CoV. And besides, they also display anti-inflammatory activity. You can see you have this first molecule, which are quite known by everyone, curcumin, which is the main substance obtained from curcuma longa, and it shows let's say significant activities against cysteine-like protease and papain-like protease, and it shows good anti-inflammatory activity with inhibition of cytokines and chemokines. His, let's say, his congener, namely CERSusChem, also show activity against these two proteins, as well as the reduced, well, it's not the reduced form of curcumin because it's quite different. You don't have the methoxy function, but CERSusChem, it shows good activity against PL-Pro and it was weakly active against CL-Pro. But it also showed a bunch of anti-inflammatory activity and inhibition of cytokines and chemokines. And the last one was oregonine, with good activity against papain-like protein and weak activity against cysteine-like protease. And if you compare all these four structures, what happens is you will notice that those which has alpha-beta unsaturated ketone show activity, good activity against 3CL-Pro. But once you started losing the alpha-beta unsaturated moiety, there was a decrease of the activity against cysteine-like proteins. And the PL-Pro is, this is a point of view, is for me, it seems like it is more related to the phenolic function, because all of them show quite a good activity against the PL-Pro. So, and you can see that all of them have this phenolic moieties. So this is the first thing I was able to observe as a structure activity a relationship. There were also some triterpenes with quite good activity against these proteins. The first one is cholesterol with a moderated inhibition effect, and the second one, let's say the methylated form of this molecule with a significant activity, and betulinic acid, a moderated activity. His activity was quite comparable with the one of cholesterol. And beta-sitosterol show, let's say, weak activity. But which is interesting is all of them are anti-inflammatory agent, and cholesterol show his anti-inflammatory activity against cytokines produced by human lung cells. And of course, we can also highlight here a kind of structure activity relationship. As you can see, the methylation of this acid function increase the activity by twofold. So this is also something which is quite interesting. And then among all these compounds that I presented, some of them was able to have three biological functions: inhibiting the protein, showing anti-inflammatory activity, and modulating the immune response. So you can see them. You have apigenin, which is a or let's say all these 5 molecules or 6 molecules with curcumin are all from food. So we have here once again a good point that can be explored. And these 2 diterpenes was also able to modulate the immune response, besides showing the inhibition of effects against the protein and anti-inflammatory effect. After collecting all this data from the literature, we decided to run some in silico tests. This was performed by collaboration with Professor Marcus Scotchy. So all this 79 compound was submitted to in silico study and using different consensus docking analysis, different model. And both the target which was used here was PL-Pro and 3CL-Pro of SARS-CoV-2. So we used all these molecules which were found in the literature with inhibitory effects against, let's say, cysteine containing protein from SARS-CoV. We use them to build our in silico model. And after the whole study and interpretation of data, we found that amintoflavone was the, let's say, the leader of the group. He was the compound with the highest score as a drug leader. And here I can show you some picture with the amintoflavone in the pocket of a cysteine-like protease. And you can see that here we have some interaction with cysteine. Later on, I will perform a discussion to indicate why maybe we have this kind of interaction. So it fits very well in this pocket with cysteine near to these two aromatic cysteine. And so, why this model can be efficient? We also check in the literature the relationship between all these inflammatory mediators with COVID-19, and we found out that TNF-alpha, IL-1 beta, which are interleucin, and tumor necrosis factor alpha, where all pro-inflammatory mediator found in multiple cells of patients suffering from COVID-19. This is the first thing. So, if amintoflavone, apigenin, and epiritin can inhibit all these cytokines, maybe it's a good starting point to explore drugs against COVID-19, because they are able to inhibit the main protein and other cysteine containing protein. They are able to reduce the inflammation, and so I guess this is a good. And in silico study shows that amintoflavone is a heat compound, so this is quite interesting. And then another literature search shows that clinical report shows that the part of hospitalization case of COVID-19 have a respiratory problem with the possibility to undergo progressive and severe cardiovascular damage. As you remember, I highlighted those structures when I showed on some slides before. I highlighted them because they were able to reduce the inflammation in cells related to lung and heart. So this molecule also can be used for drug discovery or as a functional food. And I guess maybe we will have some response. And another point which is really important with infectious diseases is that N-oxide species are the first line of antimicrobial defense produced in living cells. But once we have infection, what happens is the cell will produce N-oxide and has a chemokine. And this N-oxide has some function. The first function is antimicrobial because it stops the microbial replication. So the second activity of N-oxide species is to reduce inflammation, vasodilatation, and anticoagulant properties during the infection. But there is a problem. If N-oxide species are overproduced, what happens is they will cause damage such as circulatory failure, septic shock, tissue damage, inflammation, and cytotoxic reaction. So there is a good point here. If we found a natural product that can inhibit or mitigate N-oxide production, this could be really relevant to, to alleviate the overexpression of this chemokine. And another point that I wanted to bring here is, okay, we are testing a lot of compound against cysteine containing protein. Why? What is the point of doing that? This page is, let's say, we are dropping all the molecules and looking into the enzyme. As you may know, cysteine, which with active site, enzyme with active site cysteine residue rely on thiolate, which means you have a thiol group which loses proton by the protonation and form a thiolate, and this thiolate can be really reactive. And if it is reactive, which kind of reaction it can be in which it can be involved? First, it can inhibit through electrophilic or oxidant irreversible and reversible reactions. So, which means a thiolate can react with any electrophilic site or electrophilic substances. And then, if this can happen, maybe that is why we were able to observe in the case of diaryl heptanoid that some activity was quite good because of the alpha-beta saturation, and others was quite low because of the loss of this alpha-beta saturation moieties. So we have the thiolate which are able to react in the irreversible or reversible manner with electrophilic site. And we also know that some of the polyphenol that we consume can be, let's say, can go through a metabolism, which and this metabolism can produce, let's say, can oxidize those polyphenols, and so the electrophilic character of our cysteine protein can be by indirect reaction or by direct reaction. Why I'm calling it indirect reaction is because when you consume the polyphenol, you can directly have a reaction. What happens is it will be metabolized, and when it's metabolized, for example, catechol moiety will form an orthoquinone, and hydrobenzoquinone will form a benzoquinone. So once you produce this scaffold, what happens is you can see that I highlighted here in red the alpha-beta saturated system in both substances. And then I also chose some of the compound which show good activity, such as flavonoid, and here you can see the same system, and the triterpenes, the quinone triterpenes, you also have the same system. The diterpenes, you also have the same system, and the diaryl ethanoate, you also have the same system. So, which means if you have substances with this system, they can be really versatile prototypes to develop cysteine protein inhibitors. And then, just to resume, you have cheolates which can attack electrophilic substances, and then they can form after that they can form a covalent bond, and then once this happens, at the beginning you have a protein which is one of them which assume or which is responsible for a main let's say role in the virus replication. And then you mix this virus with a substances. What happens is you have this kind of reaction going on. So your protein will bind with your substance and form something else. And this something else is not recognized by the biological system of the virus. So what happens is at the end you will impair the virus replication. And this is what this page is meaning. No? The entire process impair the enzymatic activity because you have a new structure and a new configuration for the protein, which is unknown by the biological system of the virus, because it's a new substances which has nothing to do with the living cycle of the virus and the protein of the virus, which react together by let's say an electrophilic addition, and then you form a new complex which has nothing to do with the virus, and consequently this inhibits the replication. So that is why we believe that cysteine protein has a proximity with this two systems, because if you have oxidation or let's say metabolisms of this flavonoid, what will happen is you will have a methyl system here, which is a kind of quinone with double bond carbon double bond here. And then it's become, and then you will form a kind of alpha beta unsaturated ketone, which is a good electrophile or which can readily react with cysteine protein, and then you will have the inhibition of the virus. Maybe that's why allantoflavone is a good candidate. And on top of that, you also have different protein which are really close to this site, and so maybe that is why he's a good candidate to explore drug discovery against COVID-19. So in conclusion, 79 metabolites were found with potential antiviral activity on the SARS-CoV-2, and they are all endowed with anti-inflammatory properties, and among them 14 can modulate the immunologic response. And this molecule belong to the family of flavonoid, chalcone, xanthone, diterpenes, aryl ethanoate, alkaloid, catepenes, amide, and lignin. And what I'm going to include is this work have been submitted as a publication or for publication. So it has been accepted. Now we are just running some last correction in order to finalize the whole process of publication. So there are more substances, and as I said before, I couldn't show all of them because 15 minutes is not. I think we'll spend more time than 15 minutes. And then the mechanism of action of this natural product could be through irreversible microaddition, as I explained, and direct reaction between the protein and the compound, or indirect reaction occur after the metabolisms of the natural product. So, which means this microaddition can occur directly or indirectly just after the metabolisms. So I cannot finish without thanking my colleague, without sending them some acknowledgement to Professor Braga, because he was part of this work too, and Professor Francisco and Professor Matos, which he was responsible for the in silico test, and we also thank CAPES because they give us some financial support for this project, and we are nearly, let's say, four or five professors working on this project. So I guess this is the end, and I thank everyone for his kind attention, and I'm willing to give you some response if you have some let's say, if you have some questions. Thank you, everyone. There is a first question here from Thais. Thank you, Thais, for your participation, and the question is: it is possible that the alpha beta unsaturated structure can be involved into a micro-irreversible system of inactivation of CL-Pro and PL-Pro, just as happened on M-Pro. Thais, this is what we are thinking. I guess it's possible, and actually, I can really tell you about the selectivity, because we will take the system of curcumin, for example, where we have some phenolics and the alpha beta unsaturated system. So since the cysteine, or let's say the thiol containing protein can do this attack at the beta position by microaddition, I guess this is possible, but now I don't know if CL-Pro or PL-Pro where they prefer. Maybe it depends on the pocket. Where are the approximation, you know? And then if the aromatic system or if the phenolic system is more near to the cysteine residue of the PL or the CL, maybe they will attack the aromatic system once it's oxidized after the metabolism. But maybe if you also have, I guess if you have the alpha beta unsaturated system, this is possible. And what we are about to do with some collaborators, and I found also some literature about this, is maybe we are going to prepare two systems. A system which has the alpha beta unsaturation, and a system which has only the phenolics, but the phenolic will oxidize it to have a benzoquinone prototype, and then we can mix them with cysteine itself and see what happens. Maybe and see which one will be more reactive. What is the let's say the kinetic behind this? I don't know if one is more reacting than another, or maybe it's more. Maybe when you have the alpha beta unsaturation system, is ready to be react, and then the other one spend more time, and maybe that's why you can have a difference of activity. But I guess the alpha beta unsaturation system is, let's say, it's a good prototype for this, for the development of this kind of drugs. The toxicity, actually we didn't we didn't run any toxicity yet. What we are doing is, I know some of the compounds. There's literature about the toxicity. But I know quercetin, camphorol are less toxic. This is what I know, but it's quite different if you have the pure single molecule. It's quite different. Maybe the toxicity can rise. This is something we are going to check, because actually we are working with all this process. The main thing we are trying to do is isolate the compound, run some toxicity tests, and then try to modulate the compound and see what can be improved. But I guess this will be at the end. But I have no data about the whole toxicity. But the only thing I can say about all these compounds, those which are from food, until now there is no toxicity. Let's say they are not quite toxic, but the quinones, such as these diterpenes with orthoquinones, I found something about allelopathy. So they are quite toxic compound, because they show they have good cytotoxicity, and some of them are quite unselective. They can affect normal and tumoral cells. This is what I know. And also some quinones. Some quinones was reported to show allergic reaction, but there are also some studies showing that if you block some, if you can put some, let's say some functional group at the alpha position of the carbonyl, which is in the quinone nuclei, and there are some function if you are able to introduce in your scaffold, you can reduce the toxicity. But overall, the food, the molecules which are isolated from food, they are really less toxic. This is what I know. And those which are not, which are from medicinal plants, I guess there is a long way to be made before, let's say, taking any decision. So I don't know if I give you the right answer. Another thing that I can show, I found epirichin. Epirichin itself has a supplement, if I remember well. Epirichin itself has a supplement, diazein too. Because I guess if they are selling as a supplement, it's because the toxicity is not, even though they can recommend to consume not more than, let's say, not more than 1 pill per day. But I guess it's not, they are not really toxic. Obrigado, Caroline, pelo convite e tudo. Foi um prazer, a primeira vez que eu estou apresentando assim, mas parece aula. É bem interessante fazer isso e agradeço muito pela experiência, porque é uma boa experiência para mim. Então, qualquer coisa, a gente está aqui. Muito obrigado para tudo, agradeço muito. Se você tem o meu contato, se alguém tem uma dúvida ou alguém tem uma curiosidade sobre o projeto, dá para discutir, ou colaboração também, porque acredito que esse tipo de convite, o mesmo de apresentação de palestra, é para estabelecer as colaborações e aumentar o seu horizonte. Então, sou eu que agradeço.